Hi guys, again welcome you in the YouTube channel of Chamila Devi Institute of Pharmacy. In order to understand the interaction of drug molecule with the respective receptor, we are going to study the various structure activity relationship in our previous discussion, and now we add in that the structure activity relationship of cholinomimetic agent. If we look for cholinomimetic agents, the compound which mimic the action of acetylcholine is called cholinomimetic agents. Here the acetylcholine is showing their activity on two different type of receptors, and definitely if we look for the pattern how they bind to these receptors, you must aware about the structure of receptors, the hot spots which present in the receptor, and how they are crucial for selectivity towards the specific receptor. So if you look for cholinergic receptor, there are two type of cholinergic receptors. The first is known as nicotinic receptor, while another is known as muscarinic receptor. And actually this name come in the picture on the selectivity of these receptors towards the natural product. Nicotinic receptors showing selectivity towards the nicotine, while the muscarinic receptors showing the selectivity towards muscarine. But actually the acetylcholine is a natural messenger for both receptor type. They are unable to differentiate in between the nicotinic receptor as well as the muscarinic receptor. Now the question arises, why this acetylcholine is able to interact with this two type of receptor in a non-selective fashion. So before that we must understand the subtypes of these receptors. So if you look for the muscarinic receptors, these are receptors further classified into 5 subcategory, that is varies from M1 to M5, and all these receptors are G protein coupled receptor. While if you look for the nicotinic receptor, these receptors are two types, that is known as N1 and N2, and nicotinic receptors are ligand gated cation channels. So for these two receptors, the muscarine is a agonist for muscarinic receptor, while nicotine is agonist for nicotinic receptor. But the acetylcholine is the natural neurotransmitter for both of the receptors. So we must aware about the natural ligand which bind on the cholinergic receptor. For that, we take a look of the structure of acetylcholine. So here the structure of acetylcholine, and if you look for this structure, it mainly having two parts. One part is called acetyl segment and the another one is called the choline segment. So when these two parts are get combined, then it give acetylcholine, and we divide the acetylcholine into three key zones. The first segment is called ester segment. The another is called quaternary amine segment, and the third part is called the linker, which present in between the ester moiety and quaternary amine moiety. So now, after knowing the structure of acetylcholine, the next thing which come in the picture is what is the probable site of binding of this acetylcholine. But now we are having a much more information about the binding of acetylcholine with the acetylcholine receptor. So we take the look of receptor, that is look like this, and generally in receptor there is cavity which is required for dock of the molecule in that specific zone, and here this is the cavity of the receptor, and in this cavity few hot spots are there which is crucial for the various interaction of ligand with the macromolecule. So what are the hot spot in the cholinergic receptor? If you look for cholinergic receptor, then there are the various hot spot like that. The first is asparagine is there, then the other one is the anionic zone, that is come through or generated through aspartate, and some are hydrophobic areas, the hydrophobic pockets where the interaction is occur. So if you look the structure of cholinergic receptor, in which the one hot spot in the form of hydrogen bond donor and another hot spot in the form of anionic zone, and rest of the part is in the form of hydrophobic zones or hydrophobic pocket. And this hot spot decide the interaction of this macromolecule with the receptor through their complementary actions. Means suppose if there is the hot spot in the form of hydrogen bond donor, that is the asparagine, then definitely the complementary part in the ligand is must be the hydrogen acceptor. So if you look for complementarity, then there is the complementarity come in the form of hydrogen acceptor is complement to hydrogen donor. Okay, then similarly the receptor having a anionic part, then their complementary part is on the ligand, that is cationic 1. Okay, and if you look for the other hot spots, that is hydrophobic zones which present in the receptor. So again, for complementarity in the ligand, the hydrophobic group are moiety, which are complement to this hydrophobic pocket, and they undergo for interaction in this area. So ultimately, when we look for the receptor, they suggest that there should be the cationic portion on the ligand, there should be hydrogen acceptor on the ligand, and apart from that some of complementary hydrophobic groups are moiety, and generally these are located in the three-dimensional space. So the orientations is also crucial, as well as the distance of the complementary part is also important. Means definitely if you look for hydrogen acceptor, it should be having some crucial type of distance from the cationic part. So if we keep in mind or we fulfill all this basic requirement, then the molecule is able to fit properly in the cholinergic receptor, and they produce the necessary conformational changes which create secondary responses. There is another possible reaction which may occur if the ligand having that type of group or moiety which is complementary to the aromatic portion of the receptor. That is the induced dipole interaction which take place in between the aromatic ring of the tryptophan of the receptor and the cationic portion is present on the ligand. So this is called induced dipole in the aromatic ring. These are the possible reaction which take place in between the ligand and the macromolecules. If you look for the three-dimensional structure of muscarinic receptor, then it looks like this one, where the different amino acids which are showing here, they are present on the active side, and this is the 2D representation of that active side. So now we know the two type of cholinergic receptor. One is called nicotine, another is called muscarine. Then how this acetylcholine is able to bind on both the receptor, while we have the pure agonist for muscarinic receptor as well as pure agonist for nicotinic receptor, which are quite differ from each other. So for understanding of the undifferentiation nature of acetylcholine towards both of the cholinergic receptor, we must know the basic structure understanding of acetylcholine. If you look for the structure of acetylcholine, then you see it is a straight structure in which the single bonds are there, and this single bond is freely rotatable, and due to freely rotatable nature of this single bond, this acetylcholine is present in a large number of conformations, in which the angle which present in between the methyl and carbon may vary from the 1 to 0 to 360 degree. Similarly, the angle in between carbon to oxygen is vary from 0 to 360. Similarly, this angle is vary from the methyl end to the quaternary nitrogen end, and that's why there are huge number of permutation combination orientations of this group or moiety in the three-dimensional space, and that's why there is the multiple conformations are possible for the acetylcholine, and in this multiple conformation the distance is also vary. If you look for the distance in between the oxygen of ester to the nitrogen of quaternary amine, they are changing, and these changes might be responsible for the non-specific behavior of acetylcholine towards the nicotinic as well as muscarinic receptor. If we look for the different conformers of the acetylcholine in their Newman projection, here I took the key conformer. One is synclinal, that is might be known as gauze or isque, and if you look for the difference of the nitrogen of amine to oxygen of ester to the synperi planar conformer of the acetylcholine to anti-cleaner conformer of acetylcholine to anti-conformer of acetylcholine. This difference is in two-dimensional space, still it is identifiable. There is the distance is changed from synclinar to synperiplanar to anticlinar to antiperiplanar, and definitely these variations as might be responsible for non-specific nature of acetylcholine. In one conformations they might bind with the nicotinic receptor, while in other conformations they might bind with the muscarinic receptors. So this synperiplanar is eclipsed, where the quaternary amine completely cover the oxygen portion or this carboxylate area of this acetyl group, while in case of anti they are completely opposite. So this eclipsed form is also known as cissoid, while if you look for anti, that is called transvoid. For better understanding of this, we try to study the molecule in which free rotation are hindered. In acetylcholine, free rotation are not hindered because it having a single bond. But if we study with the compound in which the free rotation are hindered, then it give better understanding of the specific or specificity of the cholinomimetic agents. So, if we look for the both receptors, that is the muscarinic receptor or that is the nicotinic receptor. In case of muscarinic receptor, the distance of quaternary amine nitrogen to the oxygen which are able to form a hydrogen bond is 4.4 angstrom. While if you look the distance which bind with the muscarinic receptor, then it is 5.9 angstrom. And this is only feasible if there is the possibility of the different conformer in which the distance of quaternary nitrogen to oxygen is vary, and that is feasible in case of acetylcholine. But if we hindered the rotation, then this distance is remain fixed, and in such case the molecule are able to bind with the muscarinic receptor or might be bind with only the nicotinic receptors. So, if we look for the rigid analogs of acetylcholine, so there are few of the rigid analogs in which the rotatable bonds are locked within the ring. This ring hindered the free rotation around this, this or this bond or this bond. The same thing happened here. But if you look for ACTM, which is synthetic compound in which the cyclopropane are there, and this cyclopropane, although they have the single bond, but it is cyclic structure, and that's why the rotations are hindered. They are not freely rotatable, and that's why this ACTM may occur into different conformations, which is the trans conformer in which the acetyl moiety and the quaternary amine group are opposite to each other, while it may also present in the cis form. But these two forms are not interchangeable. Means this quaternary amine groups is lie on the same side, then it is known as cis geometry. So in this case, the space which present in between the quaternary amine nitrogen to oxygen is very as compared to the cis geometry. So if you look for the cis and trans form of ACTM, this is rigidly constant to the conformer. Like here, if you look for the cis isomer, that is similar to the syn periplanar geometry of acetylcholine. And if you look for the trans ACTM, which is having approximately similar conformation as anti-cleaner geometry of the acetylcholine. choline. And if you look for look for the potency of the trans geometry as well as cis geometry of ACTM, the trans enantiomer is equally as or more potent than the acetylcholine towards the muscarinic acetylcholine receptor. While if you look for the cis-ACTM, the racemic cis-ACTM compound had almost no activity for muscarinic acetylcholine receptor because this thin periplanar having a freedom for rotation around the single bond. But if you look for the ACTM, in ACTM, basically we restrict the rotation due to presence of this cyclopropane or this cyclic structure restrict the rotation, and that's why this geometry can't be transferred into the trans geometry, and that's why the trans enantiomers showing the activity towards the muscarinic acetylcholine receptor, while cis geometry is unable to show the activity towards the muscarinic acetylcholine receptor. Means ultimately the distance which present in between these two groups, that is nitrogen and oxygen, is changed. Basically we require the distance that is 4.4 angstrom, but it might be shorter than that, and that's why they are unable to do the action. And similarly, if you look for nicotinic receptor, then all the compounds of ACTM, which might be cis enantiomeric form or cis geometry, trans enantiomeric form or trans geometry, they are very weak nicotinic agonists because the distance is still not reached to 5.9 angstrom, and they are not showing the binding towards the nicotinic receptor. So these things help in designing of the selective muscarinic receptor if we restrict the rotation by means of incorporating the cyclic system to the molecules.